Okay, so we have already lots of attendees, so I think I can start. Welcome everybody to the DFHCC Seminar Series Connect Science. My name is Judith Agudo, and I'm an assistant professor in the Cancer Immunology and Virology Department at Dana Farber Cancer Institute. And it is today my great pleasure to introduce to all of you Dr. Alexandra Chloe Villani. Dr. Villani obtained her bachelor's and PhD at McGill University in Montreal. My French is not that great. In Canada. And after that, she came to Boston for her postdoctoral training with Dr. Nir Hacohen, where she did really outstanding and super interesting work with single-cell technologies studying human immunology. In particular, there is one paper that I like a lot, her science paper describing dendritic cells and monocytes in blood from humans, which I think was really, really nice in 2017. And after that, she started her independent lab at MGH, where she has continued this deep dive in human immunology using single-cell technologies, and with a major focus in immunological adverse events happening during immunotherapy or immune checkpoint blockade. So without further ado, Dr. Villani, thank you so much for accepting the invitation, and we look forward to your seminar.
Thank you so much for such lovely introduction. It's my great pleasure to be here today and to share with you some of the recent progress in our work. And so let's see if I can move forward. So to keep it brief, since it was so properly introduced, the lab does indeed study immune-related adverse events, or IRE, as I'll be referring to the use of immune checkpoint inhibitor therapies, as we strongly believe that solving the underpinnings of IREs will ultimately help us improve the success of design of treatment. And I hope the following slides will convince you for the motivation behind this work. So to this date, we have over 233,000 cancer patients in North America that are eligible for these drugs that are used in now more than 57 different cancer types. The Achilles heels of these treatments is the development of such IREs that can truly affect any organ of the body, anywhere from heart inflammation to neurotoxicity or simple skin rash, and the incidence is continue rising. Here you have the number of patients at MGH that have been admitted with IREs. So these are the most severe patients. And on the right, you have the breakdown of the different type of IREs we see at MGH, with, of course, the greatest portion being involvement of the colon, followed by the liver. But truly, we capture the full spectrum of immune-related adverse events. And so some of the main challenge in the clinic is that everything is reactive, nothing is proactive, right? And there's no solution that currently exists for timely detections of adverse events. Current solutions include withholding or stopping these life-saving immunotherapy treatment, or using broad immunosuppressive solutions such as steroids, which can ultimately affect anti-tumor treatment efficacy. And we expect that the IRE incidence will continue rising as immune checkpoint inhibitors are becoming standard of care and now combined with other treatment modalities. There's a need for better and more rapid diagnostic tools. IR presentations in the clinic are really hard to diagnose. It's hard to know if you're dealing with opportunistic infection or progression of cancer. We don't have any biomarkers. It's actually hard to identify patients that are at risk for developing IREs. And ideally, we would have some so we could stop the treatment before these adverse events become irreversible. Because unlike chemotherapy, when you stop the treatment for chemo, the side effects may stop. It is not the case for adverse events. And as I mentioned, there's a need for better targeted therapies beyond the simple use of steroids, because ultimately this can affect your anti-tumor immunity. There's a challenge for studying IREs in animal models, because most IREs are not recapitulated currently in currently available mouse models. So there's a need to study them directly in human subjects. So what we've built, and I'm sure some of you might have heard of this service at MGH, together with my partner in crime, Dr. Kerry Reynolds, we've built this translational program that sits over the severe immunotherapy complication service, where we have different oncologies across different cancer disease center, including melanoma, GI, breast, head and neck, thoracic, that work towards identifying patients in an inpatient setting with likely IREs and coordinate their care with over 51 members across 6 departments of and divisions of 10 divisions of medicine. So every line is a different specialties. And these is the wheel of care. These are specialists that will see these patients and will help tailor their treatment. And so as part of this wheel of care, the translational team, which are members of my lab, they bridge between this clinical effort and the lab, and several physicians, scientists specializing in different fields of medicine, so such as rheumatology. Today, I'm going to show the work of Dr. Molly Thomas, who's a gastroenterologist attending now at MGH, working in collaboration with Mike Dugan, Danielle Zata from cardiology, Michelle Rinkarjan from endocrinology, Stephen Blum and Lady Zubiri from oncology, for example, along with computational fellows, research associate and CRCs that bridge between the clinic and the lab. So we collect tissue as part of the clinical diagnostics if patients consent to it. We don't collect as part of research. So the way we frame our strategy is as follow. We try to figure out if the different IREs involving different organ systems are the same disease and that should be treated the same way, or different entities. So to answer this question, we collect biopsies at the time of diagnostic. We try to derive the cellular network involved in driving disease using single-cell multi-omics strategies. And we try to infer pathogenic cell circuits and cell states by remapping the molecular network. So that's what we refer to scale. We also include a time component. And so in some cases, we actually have access to blood and plasma serum through our collaboration with Geneviève Bolan from the same patients before they receive checkpoint inhibitors and after they receive their treatment before they develop adverse events. At the time of adverse events, before any steroids are given, we collect blood, serum, plasma, relevant body fluids. So for example, if it's neurotoxicity, we collect CSF and a biopsy of the affected tissue. And we carry on collecting until resolution or relapse when we can. And we collect both in the inpatient or in outpatient setting. And then we do different type of measurements depending on the amount of tissue we can collect, although the workhorse is a single cell and single nuclei sequencing. We do keep tissues for spatial imaging as well. And so to date, we've collected a cohort of close to 300 patients that are across different toxicities here mapped at the top. And you can see that they're all split in case and control. So that's because we process the tissue readily after collection. And so we never know if it's going to be a true case or control until we get the pathology report. So case and controls are all cancer patients on these checkpoint treatments, with the difference that a case has a histologically proven IRE. The control, for example, could be a patient that overused laxative and so did have some diarrheas initially, but did not end up being a checkpoint colitis. And now we have biobanked over 1,700 sample type. And so we use 3 different strategies to try to map the underpinning of IREs in our phase 1. The first one is trying to figure out the cellular and molecular perturbation defining these adverse events directly in the tissue. So we're trying to figure out the guilty cell populations driving the disease. We want to see if it's purely T-cell mediated, given what we know about the function of these drugs. We don't know if there's specific transcriptional programs that are altered. And because some of these presentations do mimic autoimmune disease presentation, we want to see how similar or distinct they are from classical autoimmune disease. So we collect the biopsies, we generate our single cell data, and then from our analysis, we can derive different information. So we're going to see a lot of these maps. And so from our single cell data, we can infer cell identity here represented by shape and color, and we have control and cases. And then we can regroup the cells based on shared feature. In this case, it's transcriptional shared features, to try to compare the populations that are distinct between case and control. So here would be some of these guilty populations enriched in colitis. We can do more than that. We can try to figure out the pathogenic cell state. So most immune cells, they don't exist in a bimodal state. They're part of the larger spectrum of pathogenicity. And that's actually pretty important to figure out the window that has therapeutic value. We can also figure out the transcriptional program associated with any toxicity, as well as infer cell communication. So that's all done in the tissue. Beyond that, we try to use the T-cell receptor clone to try to understand the relationship between T-cells in the IRE lesion, the tumor, and the blood. We want to understand if we're dealing with the same cell states driving a response to a checkpoint and the IRE for examples. And we're trying to figure out if there's common rules shared between patients with the same toxicity and even those shared across different toxicity. Finally, the third program is more focused on biomarker discovery. And this is all the analysis we're doing in the blood. We're trying to figure out if there's specific auto antibodies or secret factors that vary over time and that track with the emergence and resolution of toxicity. And based on our tissue analysis, we try to figure out if we can identify, if we identified a cell state or clonotype associated with IRE in the tissue, we then look for it in the blood as a more targeted approach for finding new biomarkers. So we have different efforts across the colon, the liver, the joint, the heart. We have a new efforts on kidney and endocrinopathies, and because all efforts are done in parallel, it also enables us to do meta-analysis. But for sake of time today, I want to tell you about a project co-led by Molly Fisher-Thomas, who is now a GI attending, and Camille Solikowski, who's a computational postdoc, and this was in collaboration with Mike Dugan at MGH. And so this is our cohort. And so we had 8 healthy control and 8 patients on checkpoint inhibitors, as well as a 13 case of checkpoint colitis. Most of them were similar enough in metadata, although unsurprisingly, patients who developed checkpoint colitis had slightly more infusions of immune checkpoint inhibitors. In terms of our study design, we collected biopsies and processed them in 2 different ways. We did flash freeze some of these biopsies to enable single nuclei analysis to be able to better capture epithelial cells and mesenchymal cells, which don't survive so well tissue digestion. And we also process the tissue fresh to enrich for immune cell and did single cell analysis of the immune cells. We also fixed tissue to do some validation through microscopy experiments. In parallel, we collected blood and plasma at the same time as the biopsy, and we also generated single cell RNA sequencing together with paired TCR, BCR, as well as surface protein. And so we're gonna start with the immune cells. Unsurprisingly, we saw T cell expansion in the colon mucosa of these patients. So you can see here the 3 different categories of patients. We see marked expansion as shown here as in a representative FACS plot, and summarized here below. And this was known. This is not necessarily very shocking, and also been recently reported last year in the Elegant Cell paper. And so I want to start by showing you our analysis of the CD8 T cell compartment, which I should mention comprised a little bit over 25,000 cells. And so we uncovered 12 different cell populations here labeled 1 to 12. Every color captures a distinct cell population, and the arrow tells you if the population was up or down regulated, as depicted here through the odds ratios. And so you can see that we have, oh, my titles don't appear, I'm not sure why. You can see that there were specific populations that were enriched, and that there were some heterogeneity in these populations, where you see at the far left, the naive and resting T cell population, at the bottom and the far right, some of these effector T cell population marked by coenzyme B and K. Specific populations were enriched. The top ones include integrin beta 2, and so population 11 and population 3 were most upregulated in these in checkpoint colitis. While this box here, all of these populations are express integrin alpha E, and with several of them, including a cycling population, being upregulated in checkpoint colitis. And so I should mention, I'm sorry, my titles are not appearing, but the point to be made here is that these were likely captured different type of CD8 TRM populations in checkpoint colitis. And so I want to say a few things about the CD8 tissue resident memory TRM, or here TRM cells in the human GI tract. So these TRM cells are long-lived memory effector cells that can make up a majority of the T cells in most human organs. CD8 TRM cells play an important role in tissue surveillance and recall response to pathogens, and they do not recirculate. And you know, it's not trivial to study TRM directly in human. Traditionally, it's been done in mouse through parabiosis. You can do cellular labeling and do intravascular injections, so you can try to track these cells. There's good tissue transplantation and cell surface protein followed by transcriptional profiling studies that have been done. And there's a very elegant paper that was published last year in 2021 in a transplant model of the GI tract in human that shows that cells in the GI tract, CD8 TRM cells in the GI tract express both integrin alpha E and integrin beta 2, like the cells we observed. And so this is the model from this published paper in human transplant of the colon. And the point that I want to make here is that the cells they report as integrin alpha E, we see similarly in the cells that they express integrin beta 2, we also see similarly, and they show significant heterogeneity in our model. They show heterogeneity at the transcriptional level. So here you can see again, the middle part are integrin alpha E. They have different transcription factors, whereas our integrin beta 2 express mostly EOMs, whereas integrin alpha E, you have Hobbit and as well as these two transcription factors. Sorry about this. They also express, there's also heterogeneity in terms of the cell surface and granzymes they express. The integrin beta 2 express mostly some of these CXCR3, CR1, and granzyme K markers, whereas integrin alpha E express more granzyme B. They also have only the integrin alpha E, a subpopulation of them here, population 5, express some of these more exhausted markers. And I should say, every time I say this, these are those that are associated with checkpoint colitis. And so we asked, can we leverage T-cell receptor analysis to try to infer the relationship between all of these CD8 T-cell clusters, right? Before I get there, I guess to summarize, we see two different populations associated with checkpoint colitis. One that is with different level of CXCR3, CR1 and expressing EOM circumscription factors, and the other one that we call integrin alpha E that express Hobbit, ID3, AK, FZ2, as well as exhaustion marker. And so for those of you who don't think a lot about T-cell receptor, right, you have these unique barcodes and each associated with specific clone time. And so as you map the TCR clone over time across tissue and across blood, we can start asking if we see the same TCR clone across tissue compartment. And here, in this particular study, we're matching alpha and beta variable region and CDR3 region. And so first, I want to mention that we saw a greater diversity of TCR clones in case versus control, and greater TCR clonalities in controls. So interestingly, the population that is predicted to likely circulate because it expresses CXCR3, CR1, share very few TCR sequence here. So the way to read this heat map is the darker it is, every number is a different cluster identity here mapped to this UMAP, and the darker it is between two numbers, the more it shares TCR clone. And you can see that there's very few shared TCR than expected. And that's further confirmed here on the right. And so we, as I mentioned, CXCR3 can be a marker for circulation, but we did see several other markers of circulation expressed specifically in these integrin beta 2 high population here for further supporting our hypothesis. And so to further summarize, we see very little sharing with all other clusters, and I should have mentioned we see more sharing with the integrin alpha E populations. So we ask, do integrin beta 2 cells have increased TCR sharing with the blood CD8 T cell population? Because based on our data, we hypothesized that this would be the case. So to stay brief, we did generate over 300,000 single cell data from our paired PBMCs, and we didn't see any differentially abundant cell population or gene expression between case and control for all immune cell population. Nevertheless, we could use the T-cell receptor data to try to understand the relationship between tissue and blood. And so that's what I'm going to start showing you here. So again, as a reminder, we're focusing on these integrin beta 2 high population here, cluster 11 and cluster 3 that also express these circulating markers. And we did note specifically that these two populations share some of the most TCR with the blood. specifically in checkpoint colitis. And this is the same UMAP as the tissue. This is the UMAP from the CD8 compartment of the blood. And we found the counterpart for this population in the blood and show the reverse that these populations share the most TCR with the tissue, further validating our findings. And so these, and so here I'm showing you the CD8 T cell UMAP and showing you the proportion of the CD8 T cells expressing 6-3-CR1 and granzyme K. And so there's circulating populations have also been recently reported. And so this is to say that we're not the first one to observe this, but we're the first one to observe this in the context of immune checkpoint colitis. And so it's been previously reported that there could be a recirculating population that was 6-3-CR1, and this is this cluster 3 population that corresponds to this cluster 2 population here. And the other population that is likely blood confined is this population 11 here, which corresponds to this population 5 here that we map to each other. Then we ask, so to summarize, we see TCR sharing with blood T cells specifically for this integrin beta 2 high population, further supporting the fact that this population could be recruited from circulation. We then ask how similar were the transcriptional profile of these two populations associated with colitis. Are they very similar? Are they distinct? Here in this bar plot on the right, for every single CD8 T cell populations marked as a number 1 to 12, we map the number of genes that are differentially expressed at a full change of 1.5 and an FDR of less than 0.05. And you can see that some specific populations had more differentially expressed gene, including this cluster 5, which is the one that was most marked by exhaustion marker, was the one that was the most remodeled. Here in the box, I'm showing you in the green box those that are clusters that are associated with integrin alpha E high population, and in the orange box, those that are associated with integrin beta 2 high population. And what's to be noted here is that the population that are likely coming from circulation almost had no differentially expressed gene, whereas those that are integrin alpha E high had the most differentially expressed genes. And so when we look more carefully into it, doing differential gene expression here depicted in this volcano plot, we noted that some of the most differentially expressed genes included interleukin 26, IL-17 alpha, and 6-CL13. And this was looking at all the CD8 T cell population, and then we looked at these three specific genes across all CD8 T cell population here marked with different color code. And we noted that, again, the clusters here in the green box, which are associated with integrin alpha, here were the one that most differentially express these 3 genes. And you can see here, every dot represents a patient. Orange is a case, black is a control, and you can see that globally these 3 markers were upregulated in all cases. This was not driven by a few patients. We further validated this observation by immunofluorescence microscopy. Here on the left, you have controls. On the right, you have an example, a representative figure of a checkpoint colitis tissue section where we stain for CD3E, 6-CL13, and interferon gamma. And you can see that checkpoint colitis mucosal tissue is marked by the expansion of these 6-CL13 interferon gamma double positive T cells, so further validating the observation we made based on the transcriptional profile. So to summarize thus far our observation of the CD8 T cell compartment, we saw that checkpoint colitis is associated with an expansion of integrin alpha E high, so this population captured by the green circle, and integrin beta 2 high CD8 TRM populations. In checkpoint colitis, integrin beta 2 high CD8 T cells that are enriched in mucosal colons may be recruited from circulation based on observation of the expression of circulating markers, shared TCR clone types with the blood clusters. And we saw specific transcriptional program upregulated only in integrin alpha E populations. I want to briefly mention the CD4 compartment analysis, where we saw marked upregulation of 3 different T regulatory T cells. So here we have identified over 10 different clusters of populations. The top 3 included Tregs that were markedly upregulated in these patients. Interestingly, when we did differential analysis of these CD4 T cells, we also observed upregulation of CXCL13, specifically in one of these Treg population here that we marked as Th1 and Th17. You can see here some UMAP where we showed the expression of specific markers to orient you, and you can see, for example, that population 6 has the distinct markers for T follicular helper cells, including CXCL13, although CXCL13 was found in other cell population as well. Interestingly enough, some of these expanded Tregs had an activated phenotype marked by the expression of TNF receptor SF4, and some of these lymphoid marker cells that were expanded in ER colitis. So you see again some of these Treg population at the top, and distinct gene expression signatures across these different Treg populations. We look at differential gene expression across these different population of CD4 T cell here marked from cluster 1 to cluster 10. And you can see that cluster 9, which is one of our Treg population that is associated with checkpoint colitis, had the most number of genes that were differentially regulated at a full change of 1.5 and FDR of 0.05. And interestingly enough, when we look at genes that were most differentially expressed in this particular clusters, we noted that several of these genes were associated with a T helper 1 phenotype, such as TBX21 and IL-12. Finally, for sake of time, I'm not going to go deep in the B cells and myeloid cell analysis, but I'm going to show you some highlights of what we observe within this antigen presenting cell compartment. Surprisingly enough, B cell subsets show no population abundance difference or differentially expressed genes between case and control. We did have one case in which colitis was developed in the absence of B cells in a patient that was treated with B cell depleting therapies prior to CAR T cell treatment, suggesting that B cells are not needed, at least for the early events leading to checkpoint colitis. For the myeloid cell compartment, we identified 8 populations, 2 of which were had inflammatory signatures that were slightly enriched in ER colitis. And what most defined the biology associated with the myeloid cells was the upregulation of interferon stimulating genes such as CXCL9 and CXCL10. We did the analysis of 39 secreted factors in serum that was collected at the same time of the biopsies, and that included several markers that we found associated with colitis at the gene expression level, such as CXCL9, 10, 13, and IL-17, and none of them were associated with checkpoint colitis when we monitored them in the blood. And it could be a question of us needing to increase our sample size, but there was no obvious signal. I'm going to spend a little bit of time telling you about some of the biology we learned in the epithelial cell compartment here. And so we observed over 23 different populations across 81,000 epithelial nuclei from 26 donors here that are labeled. Similarly, every population observed is color-coded. The little arrows tells you if it was up or down regulated. We organize a different cell identity here across immature epithelial cells, absorptive epithelial cells, mature absorptive epithelial cells, or secretory cells, or mesenchymal cells. And so, and here you have the odds ratios, give you a sense if specific populations were up or down regulated. So to keep it brief, here in the blue box are the populations related to the epithelial cell compartment, where we saw marked change, we saw a decrease in the stem cell population, an increase in the transit amplifying cells, which would make sense if we saw increase in epithelial cells turnover. a decrease in tough cells and a decrease in specific populations of epithelial cells. In contrast, we saw marked upregulation of endothelial cells and myofibroblasts here in cluster 13 and 21. We also, related to the mesenchymal remodeling, we saw that endothelial cells were more abundant in checkpoint colitis, and this was associated with HIF1 alpha upregulation, along with the upregulation of other vasculogenesis genes, which could suggest that low oxygen tension in the context of the colon mucosa could drive angiogenesis in this context. And so here I'm showing you an example of HIF1 alpha expression between case in orange and control in black. And we're looking at the expression across all of the different cell compartments. And when you have a significant differential expression between case and control, it is marked, denoted here by a white dot. And so one possible mechanism by which integrin beta 2 high CD8 T cells could be recruited from circulation could be related to angiogenesis. This is Scott. And so we also noted that epithelial cells in checkpoint colitis showed strong interferon signatures associated with cases and not control. So here I'm showing you the number of genes that are upregulated in cases versus control. So upregulated being in orange and downregulated in being in black. Again, with a we're denoting the genes that are upregulated at a 1.5-fold with an FDR less than 0.05. And this is summarized here through this volcano plot, looking at all the epithelial and mesenchymal cell compartment at the same time. And you can see that there's several ISG genes coming up, including STAT1 and CXCL11 and class 2 here being upregulated. We also noted that mature absorptive epithelial cells show significant differential gene expression downregulation of several genes. And so we asked, where are these cells located in the intestinal crypt? It turns out that there's been a beautiful paper published with a crypt axis core signatures here, where you have all the key genes listed here. And we noted that all of the following cells, the clusters 2, 11, 20, and 10 expressed the most of this crypt axis core, which are also some of the cells that express the most ISG signatures, which led us to conclude that the cells expressing these interferon signatures were at the very top of the crypt. And this is another way to look at it. This gene signature here is summarized here through this brown color scheme. So the more brown you are, the more you express the script signature. So you can see it's at the top right and at the top left. And you can see these are some of the cells that express the most class 2 and some of them the key ISGs. Beyond this, we also looked at other genes that were upregulated, including interferon-stimulated genes, T cell signaling, microbial sensing, and water transport. And so you have some of these examples of genes here. Every column is one of these different clusters. The more red a color is, the more a gene is overexpressed. And when it's significantly different between cases controls, as denoted with a white dot. And so you can see that a lot of these mature absorptive epithelial cells at the top of the crypts are the ones that are expressing all of these ISGs and T cell signaling, microbial sensing genes that are upregulated in patients, and they also downregulated some of these water transport genes. And so interestingly enough, I want to point out to one rare population of cells, cluster 20, that is also at the top of the crypt and seems to be downregulated in colitis. And so one question is, could IgG exert an epithelial protective effect in checkpoint colitis? And so we try to further validate this observation that IgG signatures would be observed at the top of the crypt by immunofluorescence microscopy. So here on the left, you have a control, example of a control. On the right, you have an example of checkpoint colitis patients. And when we zoom in at the top of the crypt, you can see that in checkpoint colitis, PD-L1 and likely other IgGs upregulated at the top of the crypt epithelial cells, along with CD68 expressing myeloid cells, further supporting our observation. of the transcriptional signatures. So to summarize, it looks like if you go from the top to the bottom of the crypt, we have an interferon gene signatures that go from very high to very low. In checkpoint colitis, we see increased turnover marked by relative depletion of some of the stem cells and increased cyclin cells. The checkpoint colitis epithelial cell damage is marked at the luminal surface by strong interferon genes and loss of water transport genes such as aquaporin genes. I say I'm ahead in my time and the time, but I want to mention a few things about cell-cell interaction. So we did some cell-cell interaction analysis to try to look for which cells would be expressing some of the commonly used ER colitis therapeutics. So at every column refers to specific cell-cell interaction. So whether it's between CD4 T cells and B cells or between CD4 T cells themselves, every row is a different pair of genes that are disrupted by a specific drug targets. And we could see that, for example, anti-TNF seems to be disrupting some of the mononuclear phagocytes interaction with different immune cells, whereas Vendolizumab seems to be disrupting several of these interactions driven by epithelial cells, which is further summarized here at the bottom. We also mapped cell-cell interactions targets commonly used, and some of these experimental one being used in checkpoint colitis. And so, we talked a lot at the beginning of these different integrin beta-2 high populations. And so we noted that some of these class of drugs here summarized on the right, including the S1P receptor modulators, could impact angiogenesis and some of these endothelial cell cluster 13, and also alter the trafficking of these integrin beta-2 CD8 T cells as marked here. So what you're seeing here is the relative gene expression of the genes that are encoded by the drug targets. They're organized by different cell lineage. You have the B cells, the CD4, the CD8, the epithelial mesenchymal cells, and the myeloid cells. Every column represents one of these different populations that we've described. Every row is one of these drug targets that are organized according to categories of cytokines, integrin, JAK inhibitors, S1P receptor modulators, and others. Beyond these S1P receptor modulators, we also noted that CX3CR1 inhibitors could preferentially inhibit the binding of the circulating CX3CR1 high integrin beta-2 high cells to inflamed endothelial cells. There may also be novel IBD targets that could act on epithelials, specifically upregulating checkpoint colitis, such as IF1 alpha stabilizers. So we'll try to summarize some of the key findings. And so we noted the role of two major populations. Some of these integrin alpha E TRM population present in the lamina propria, as well as some of these integrin beta-2 high CX3CR1 expressing circulating cells. And so in the context of the treatment with immune checkpoint inhibitors, it looks like some of these circulating cells may be recruited to the lamina propria. We didn't spend as much time talking about the mononuclear phagocytes, but the mononuclear phagocytes seems to be a major producers of some of these IgGs, including 6CL9 and 6CL10, which may help also with their recruitment. We noted that these integrin alpha E population upregulated in checkpoint colitis also upregulate specific programs related to IL-17, IL-26, CXL13, and integrin alpha E, and this transcriptional program may affect the function of some of these CD8 effector T cells, which may in turn affect and drive some of this epithelial cell damage that we observed at the very top of the crypt, and that was marked by upregulation of IgGs and downregulation of aquaporin. Downregulation of such cell carriers could contribute to the strong areas observed in these patients. And so it is a complex picture. Even though we didn't observe any association in the blood, it sure looks like there's a contribution of these circulating cells to driving the inflammation colon mucosa. And so I showed you an example for one of our projects on checkpoint colitis. We have many ongoing work. We have similar approach for all of these IRAs. The working horse focus on generating single-cell RNA sequencing and single nuclei data directly in biopsies collected from patients, along with paired TCR data and BCR data. I didn't mention any BCR data today simply because we didn't see any association. We infer patogenetic transcriptional program based on our single cell data. We're now adding additional data type, including looking at open chromatin to try to infer T-cell exhaustion. We're integrating more secret factors data. We're keeping immune cells to be able to do more functional follow-ups. In some individuals, there's actually, we actually have diagnosed multiple IREs, so we're trying to compare tissues within the same individuals. We're trying to validate some of our single cell data through looking at tissue organization through spatial omics and compiling detailed clinical variables. And together, we hope that with these different data input, we'll be able to build better prediction models to try to identify these guilty cells driving the disease and identify new diagnosis and prognosis targets. And so if you're interested by this translational programs, do reach out to us. We're proud to have set up this translational program that now across the boundaries of 6 departments and 10 divisions of medicine. It involves now over 51 clinicians and 20 researchers working together. And so by having biobank so many samples and by leveraging such cutting edge experimental technologies and integrative computational frameworks, we believe we may have multiple shots on translational goals, as we'll be able to compare and contrast results from the analysis of different IRA presentations. We hope we'll be able to identify predictive biomarkers for our IRAs, though we conceive with these original phase 1 studies that we'll need to increase our sample size to be able to identify such biomarkers. The goal here is to use big data to try to think about better therapeutic strategies to treat IREs while maintaining anti-tumor immunity. By scaling up our efforts, we hope to be able to inform the next generation mechanism base of clinical trials. I didn't touch too much on this, but several of these presentations, including checkpoint colitis, may resemble some aspect of autoimmune disease presentation. And so by studying these patients, we are learning about new rules of break of immune tolerance in human subjects. And so we're now integrating new single cell data from a range of autoimmune disease to understand the relationship of these IREs with classical autoimmune disease presentation, which we hope will lead to identifying novel druggable targets with new immunosuppressive potential. So today I presented some of important work led by Molly Fisher-Thomas and Kamil Slawikowski. This is all of their work and they deserve all the credit. This was of course done with Dr. Michael Dugan. I want to also acknowledge Carrie Reynolds, who's my partner in crime in this endeavor, the entire SIG service. This takes a village to do this type of work. And so we're grateful for the entire team and most importantly to the patient and their family and you for your attention. I'll be happy to take questions.
Thank you so much. I forgot to mention this at the beginning during the introduction. So please, if anyone has a question, type the question in the Q&A section rather than the chat, and I will read the question to Dr. Villani. And oh, look, now they're popping up. Okay, so Wang is asking, could you please comment more on the role of infiltrated Treg cells in this immunotherapy-induced colitis? Are they playing the protective or a pathological role? Thank you.
That is such a good question. And I don't have a straight answer. This is one of the challenge of studying human subject because you cannot completely deplete and seeing the outcome. And so we definitely see an enrichment of these Treg population, but it's hard to know if this is a consequence of, in response to the upregulation of these pathogenic program that are being upregulated by the integrin alpha E population, or if it's actually what's driving the break of immune tolerance to start with. Not sure how we can exactly figure this out. I will say that we don't have a lot of patients treated solely on CTLA-4 inhibitor therapy, which would really help figure out if we're dealing with a protective or a pathogenic role. But it sure looks like they're contributing to driving the disease. I'm sorry, I don't have a good answer, and I'm certainly going in circle right now because this is one of the challenge of human subjects. It's kind of hard to know sometimes, and it's part of a spectrum. So it could be that they could play both roles depending on the stage of the disease. I'm sorry. This is the best I can do. Although I would welcome some thoughts if any of you have some thoughts about this.
Yeah, I guess, do they express, because you said they do have expression of some activation markers.
They do, and some of them, not all of them, express Th1 programs, which would suggest promotion of some of these pathogenic programs. But there's others that have a very different phenotype. And so I think we may have both flavors, actually, of both protective and pathogenic programs present. And what would be key is figure out how to modulate the ratios of both. Perhaps that's one way to think about it.
Mm-hmm. So Jamie Heather is asking, was the matching of TCRs from the integrin beta-2 high cells in the blood done within the same donors, or were you finding the same clonotypes in the blood of different patients? As if so, were you able to find potential HLA restrictions?
That is a good question, Jamie. And we were looking at blood that were matched, just in patient collected at the exact same time. So when we collect biopsy, we collect also blood at the same time. That's why we could do this paired analysis.
So I do have a follow-up question with the HLA restriction. Is in any case the haplotype a predictive feature that you can use, or the haplotypes have never anything to do with the risk of these adverse events?
That is such a good question. And so this sample size is really too small to be able to find such genetic predictors. It's like any complex autoimmune disease, it took hundreds to thousands of patients before being able to find such association. There's been some groups at Yale that have found some haplotypes, HLA haplotypes associated with increased risk of checkpoint colitis. There's a larger ongoing effort across North America where we're all pulling our data to try to do a GWAS at scale, and it does look like we're going to have some susceptibility loci, but to achieve this we actually needed thousands of patients.
Yeah, no, I can. Awesome.
All the relative risks are really small, right, for all of these susceptibility loci.
Yeah.
So I don't think genetic alone is what explains why some patients are developing IREs and others don't. No.
And then I do have, and this might be a bit naive, you did mention this briefly at the end. So if these integrin beta-2 cells come from circulation, then these patients could potentially have autoimmunity somewhere else.
That's correct. Yeah.
So how often does that happen?
Oh, I don't have statistics, and I should. We do see a lot of patients with several organs affected. Skin is very common. Thyroid is also very common. We had quite a few cases of checkpoint colitis that also developed checkpoint arthritis. And so as we increase our cohort size, we'll be able to better define these patterns of comorbidities. Yeah.
And do you think these T cells will be the ones to blame, or it's probably many different mechanisms anyway?
I think some of these programs could be shared across tissues, but I can tell you that in the context of checkpoint arthritis, it's very different cell population and transcriptional programs that are observed. And so it seems very complex. And in the case of checkpoint myocarditis, you don't see as many TRM populations in the heart, and it does look like it's also a different mechanism. And now the question is, why is it that some people will develop heart complication, another colon, another joint? And that's a hard question to answer that we're trying to figure out. I don't have an answer for you today, but happy to come back in 2 years and hopefully with more answers.
So you clearly have a lot of work.
Yeah, unfortunately, I mean, I guess unfortunately for the patients, but we are trying to get to the bottom of it. The other angle that we're trying to push is understanding the relationship with the anti-tumor immune response, and if any of these programs are shared with the tumor, and hopefully we can find programs that are not shared with the tumor, because that would allow us to develop more targeted therapy. So for example, we noted that IL-26, CXCL13, and IL-17 co-expression is not upregulated in every tumor. It is in certain tumor, but it is not in every tumor. So that opens the idea that perhaps we should be thinking about tailored therapies for different type of adverse events.
Oh, wow, that's very cool. And I do have one last question. So I thought it was fascinating that you could find differences in the stem cell versus the transit amplifying ratio, which would probably suggest that regeneration is impaired in these patients at the epithelial layer. And I was wondering if this is something that had already been shown in the non-immune checkpoint blockade induced colitis. And I guess related to that, also the interferon signature that goes from up in the villi to down, I thought it was also super interesting. So I guess that has to do with the position of the T cells, correct?
Yes, yes, that are collocated at the very top of the crypt. So this high epithelial turnover has been reported in IBD. This is where we found this crypt axis signature. This strong interferon gradient angle, I think, is a little bit more specific to colitis, although reanalysis of IBD, there in certain forms of IBD, in certain form of IBD, we can see such signatures. Ultimately, in the case of checkpoint colitis, there's some elegant paper that was published in recent years showing the role of IL-26 being upregulated. So there are definitely some common commonalities being observed between some forms of IBD presentation in checkpoint colitis, though I will not say that it's exactly the same. There's also quite distinct program being observed. We need to do a more thorough meta-analysis actually of IBD data, single cell data set and checkpoint colitis data sets. Yes, but this is if we think about the marked epithelial cell damage, which is characteristics of checkpoint colitis, from a pathology readout, it is perhaps not as surprising to observe such high epithelial turnover characterized by these shifts in the stem cell compartment, but it is a nice sanity check to be able to actually observe it.
It's super nice that you get to see that. Awesome. So if there are no more questions, I just remain to say thank you again to Dr. Villani for an amazing presentation. This was really a tour de force, really impressive work. Thank you so much, Dr.
Thank you very much for having me.
Thank you.
Thank you.
Bye.
Bye.